how are you, my class 10 English version students? Today I am going to start a new topics. Basically, it's also very important topics. I just explained today about the genetics. Here the genetics I also explain about the Sir Gregor Johann Mendel. He also applied the genetics and also the hereditary materials. Now everyone see the books. Basically I just explained this structure is DNA. It's also RNA, protein and characteristics. Here please everyone see the books. By taking necessary signals from DNA, RNA enters into the ribosome of the cytoplasm and according to the signal, protein is synthesized. Yes, I just say this structure is DNA. It's also flow about the RNA and RNA also flow. This structure is protein and also the last topic is also the characteristics. Here's the characteristics. In case of eukaryotic cell, these proteins are also the synthesized, is first deposited in the endoplasmic reticulum and then it passes through the Golgi body and vesicle. Meanwhile, the protein undergoes many changes and is carried to necessary places. In the eukaryotic protein cell and also the eukaryotic prokaryotic cell, protein directly reaches the destination. This protein determines the nature and speed of biochemical reactions, and from there the characteristics of living organisms are determined with the respect to the environment. These characteristics include everything from the structure to the behavior of any living organisms. It can be expressed as, I also repeat this structure, basically DNA, and the next structure is RNA, and also the next topic is the protein, and finally it also goes to the characteristics. The number of genes in different organisms is not the same. In organisms of the same nature, the number of genes is more or less same. Genes are arranged in common principle alongside from one end to another end. Chromosomes are separately spread from each other in a linear form. Different replaces or the addition of the same genes can express the same characteristics in different levels. Please everyone underline this line. Basically, for example, in a pear, for tallness, the height determination gene is T. I just give the tallness gene is the T or the capital T. And for shortness, the height determination gene is small t. And this structure is the small gene and also determines it's also the small t. When T is combined, when T and D is combined, then its structure is basically tallness. The express is tallness, so T with respect to small t. Then T is respect to small t and it also the dominant gene. Basically this dominant gene is capital T and the recessive gene is the small t. Here's the recessive gene and the dominant gene. When TT is combined, it expresses its tallness. So, capital T with respect to small t is called the dominant gene and small t respect to the capital T is called the recessive gene. When in an organism two genes are recessive, only then does the recessive gene express the recessive characteristics or the characteristics. Example, only pear plant with a small t in the short stem. This structure is the small t. Again, to see the characteristics or traits of parents are expressed in the next generation or the offspring, scientist Gregor Johann Mendel did some experiments with pear plants in 1866 and discovered a valuable theory of genetics. The factor takes the presently glamorous form known as the gene. George or the Gregor Johann Mendel is called the father of genetics. Mendel took a tall and short pear plant and transferred pollen grain of a tall plant into the stigma of a short plant. In the same way, he transferred the pollen grain of a short plant into the stigma of a tall plant. This resulted in reproduction. He made sure that the other pollen grain would not appear. He reproduced seeds were sowed, and it was found that all the plants were tall in size and no plants were short. Out of these plants, one was taken into reproduction through self-pollination and seeds were sowed. It was found that there were both tall and short plants. Among them, 3 to 4 were tall and the 1/4 was short. Beneficially or the benefits of the genetics. Basically, this theory of Mendel is applied in breeding of plants and animals. The controlled sexual reproduction is done in plants and animals to bring about desired characteristics. As a result, a generation with various characteristics are produced. From this generation, desired characteristics are selected for breeding to get plants and animals that have desired characteristics. At present, this theory is applied largely for the production of improved variety of crops. Dear students, I just now explained about the genetics and also the Mendel's law. Here's the Mendel's law. I just erase this structure or these topics. First structure is the parents, two parents. One parent is the capital T and the another parent is the small t. I just assign this structure is father and this structure is mother. I just select these two structures of the two parents. Here I just remarks this structure is the two parents. Now I just give the two divided and also the gamete. First gamete is T and T. Another gamete is the T and a small t. Here, listen carefully. This structure is dominant character and this character is the recessive character. Here, I just cross on the fertilization. Here, the fertilization is also the combined, basically capital T and a small t is also combined capital T and a small t. All this structure is basically tall and is also the first generation. I just remark this structure is the first generation. Then I just select the second generation. This second generation, first structure is the T and another is the small t. Again, this structure is the capital T and another is the small t. I just do it now checkerboard. Please everyone see this is the checkerboard. I just draw here. This is the small capital T and this is the small t. Here is the father and another is the mother. I just do a T and a small t. Here's the T and the small t. I just combine capital T and capital T. It's also the homozygous and also the tall. Another structure is the T and the T. It's also the capital T and a small t, and the another formation is they are the tall and also the heterozygous. Another topic is the T and the small t. It's also the heterozygous and they are also tall. And the last stage is also the combined small t and small t. Finally, we just find the short structure. Dear students, I just explained about this structure, basically genetics, and the father of genetics is also the Sir Gregor Johann Mendel. Here I just say this is the parents, another is the gametes. This structure is the gamete, okay? And the last structure is the F1 plant or the all in tall. Please everyone write this is the structure is the all in tall. And the second generation, here's the second generation is the F2. This structure is the F2. Please everyone select this structure. 1, 2, 3. They are T, T, and the another structure is the T small t, T capital T, and small t. All they are tall. And the another structure or the last structure is the small t and small t. They are short. And then finally I just explained this structure is 3:1. Yes, this is also the very famous hypothesis or the Mendel's law. That's enough. Thank you. My dear students, in the next class, I just explain about the DNA replication. You know about the DNA replication. It's also the mother DNA and also the next generation DNA. And our next topic is the fingerprint of DNA or the DNA test. It's also most important topics for your final exam. And the next experiment is the determination of human sex. And this structure is also the very important topics, and also the genetics disorder. Here's the two structure, basically colorblind or color blindness. Another structure is the thalassemia. Then I just explained this structure. Basically this structure is the Punnett square. Yes, it's also the mathematical term. Please everyone learn it very carefully. And the next structure is theory on evolution of organisms. That's all, my dear students.